All right. Assalamu alaikum, everyone. My name is Feras Namour. I'm a third year medical student. And today, inshallah, we will be discussing the foundational histology. And we're going to cover 6 lectures of your foundation histology cells 1 and 2, epithelium, and connective tissue. Before I start, can everyone hear and see me properly? Like, is my screen visible and can you guys hear me? We're good? Tamam. All right. هلا before we start, you guys may haven't been familiar with the PAL structure. هلا the way we do PALs is we take your lectures, the specific lectures you guys take, and we presentation from them. So مثلا the resources that I've been using are Dr. Khal's slides entirely. And this whole PAL is tailored specifically to his style and what he focuses on, all right? So inshallah, by today, we will be covering cell 1 and 2, epithelium 1 and 2, and connective tissue 1 and 2. And inshallah, everyone will benefit from this as much as possible. If you guys have any questions, feel free to ask whenever. And let's begin, starting with the cell. طيب, هلا, where should you start when you study histology? First of all, histology is this new discipline to you guys. You may have never studied it in high school, but essentially, histology is the study of tissues. Now, what is tissue? Tissue is a group of cells, and these cells they are structured in a similar way that help them all perform a similar function. So if you remember your organization, the way our bodies are formed, we have cells that make up tissues, and then the tissues that make up organs. So before we can discuss histology and talk about tissue, let's first briefly run through the structures of the cell. Now, obviously, none of this is to scale, so yeah, let's begin, inshallah. Histology is very, very unique. In order to understand it to your best ability, it's always important to link function to structure. Tamam? I'm not going to explain the function to you guys. I'm not going to explain physiology, but it's important to note what the function of the thing I'm looking at. So it will make me understand why it's formed and made that way. So when we're talking about the cell, the smallest unit that makes up a person, we should know what functions are carried out by the cell. So the cell is responsible, like the functions of the cell are protein making, ATP making, lipid metabolism, and obviously the cell should live, clean itself up, and reproduce mitosis and then eventually die. Do all of this, it does it through its structure. So we have specific organelles that carry out different functions. Cellular membrane, ribosomes, all of these, we're going to discuss about them. We're going to talk about each one of them individually. But for now, it's important to understand the functions, know them, so that we can correlate it to the structures. The cell, we can split it into 2 compartments, the cytoplasm and the nucleus. All right? Let's begin with the cell membrane, our first structure. What's the function of the cell membrane? Well, you guys, I believe you guys studied it in physiology, so I'm not going to lecture you about that. But it's important to note that the functions of the cell membrane is to maintain an ion gradient. It's a physical barrier, and it's a selective barrier. And also it has membrane proteins. All right, what am I talking about? A membrane, what is this? Essentially, your cell, it is composed out of many things, right? So inside of these things, like you have your organelles, your nucleus, your mitochondria, and so on, if there's nothing wrapping it around and holding everything in place, it's going to be floating around, and that's no good. My cell membrane is covering everything and confining it all in one place, and that is the borders of my cell. هلا, my cell membrane isn't just a border, it carries out these functions. And the way it does that is through its structure. So the structure of my cell membrane is made out of phospholipids, cholesterol, and extra add-ons that we're going to discuss. Let's briefly talk about phospholipids. You guys should know this from high school. Phospholipids is basically a organic molecule that is composed out of a phosphate group, a phosphate compartment, and a fatty acid lipid compartment. So we're going to have the phosphate group, and you guys know from your organic chemistry that it's soluble, it's water liking, it's hydrophilic, and we're going to have my fatty acid group, which is not water liking, it's hydrophobic. So my phospholipid has this duality to it. It's hydrophilic on one end and hydrophobic on one end. So when I pair it up in my cell membrane, I'm going to form a bilayer, a phospholipid bilayer. And I'm going to have my phosphate heads facing the water and my hydrophobic fatty acid facing away from the water. So over here, I can see the bilayer of phospholipids. I can also see cholesterol spread out within the fatty acids. Tamam? So this is the structure of my cell membrane. It's a phospholipid bilayer with cholesterol spread out through it. And also, I have add-ons. Here I have a sugar, a carbohydrate. And I can have proteins, and I can have lipids and other things. Tamam? So all of these add-ons, whenever they form this layer on top of my cell membrane, I'm going to call this the cell core, the glycocalyx. تمام؟ هلا my cell membrane isn't rigid. My cell membrane is very dynamic. It moves around, it responds to its environment. It's like this very wavy structure. So we call it the fluid mosaic model. It's not rigid, okay? And another thing to note is that all of these structures can also move within the cell membrane. My membrane proteins, they're not bound in place. If I have a protein on this end, it can move to here or even go in like on the opposite end. All right. So this is the structure of my cell membrane, and I linked it to the function of my cell membrane. Halla, when we talk about the cytoplasm, we have obviously the organelles that are found there. We can like classify it into membranous and non-membranous. And I think you guys are all very familiar with them from high school, alhamdulillah. So we're gonna fly through it. But before this, let's briefly again discuss the functions of the cell. So we said the cell is responsible for protein making, ATP making, lipid metabolism, and obviously the cell cycle. All of my organelles here, each one of them, they either help out the other or is responsible for one of these functions. You guys have an idea about this, so let's go quickly through it. Halla, briefly, this is just for your info, by the way, but let's talk about protein synthesis. You guys will, I believe, take all of this in very, very detail in your mole lectures. So this is not really our focus here. It's just for your info to link the structures very properly. Protein synthesis, we have DNA in the nucleus that then gets transcribed into mRNA. My mRNA then has to be translated into my protein. What does that? My ribosomes and my rough endoplasmic reticulum. Once I have my protein, then I need to package it. And that is the function of my Golgi. I hope you guys have a good idea about this from high school, but if not, don't worry. Inshallah, your more lectures will cover it to detail. And here we're only going to touch upon it very briefly. So let's start with the nucleus. This is the biggest organelle in the cell, and also it's the most important one. Well, obviously it's membranous because it is enveloped in a membrane. Now the functions of the nucleus, it's the command center of the cell. All the metabolic control. It can tell me to make ATP, it can tell me to make lipids, protein, and so on. And also, it contains DNA. What is DNA? My protein coding database. Now, when we look at the structure of the nucleus, it's a bit more complicated because it's enveloped and it has two membranes. So firstly, it's a membranous organelle, so it has a nuclear envelope. All right, the membrane of it, we call it the nuclear envelope. And obviously, it's selectively permeable barrier, because I don't want anything to come into my nucleus and out. I need to be very strategic about what I want coming in and out. It's a very sensitive place with very sensitive information. Now we have two membranes covering my nucleus, and one is on the inner side, then we have the perinuclear space, and another one on the outer side. We're going to have a better image later on, and you're going to understand it better. But for now, understand we have two membranes. Between them is a perinuclear space. Now, the endoplasmic reticulum is continuous with the outer layer, and we're going to talk about that later. But over here, this is your first ever histology slide, and this is how cells look. So basically, you can see borders here. You can see a circle, and then inside it is a smaller circle. Now, this larger border over here, sorry. Whoa, what happened? Sorry, guys. All right. So this larger circle over here is my cell membrane, and then the smaller circle over here is my nucleus, and over here is my nuclear envelope, the membrane around my nucleus. Obviously, I can't see the two layers because it's very small. So It's not seen under a light microscope, it's seen under an electron microscope, but it's not really to our, it's not really our discussion for the day. Now, my nucleus, we said that it is selectively permeable. How does it do that? We have pores or channels throughout my nucleus. And through these pores, I'm going to allow certain things in and certain things out. Okay, what do I want to send out from my nucleus? Well, mRNA. And what do I want to send into my nucleus? The things that make up DNA and also ATP and so on. Tamam? This happens through the nuclear pore complex. Now it's assembled into inner and outer rings, but for the most part, you just need to understand that it interacts with the transcription machinery. Now, the most important part of the nucleus is the nucleolus. So inside this small circle again, so we have the cell membrane, and inside the nucleus, we have foci, or like small focuses of DNA. And this is what we call the nucleolus, all right? So the nucleolus, its function is the primary site of ribosomal production and assembly. Now, it's well developed in active protein synthesis, and it has two kinds of DNA, euchromatin and heterochromatin. You guys will hear plenty of this in your mole lectures. In histology, all I want you to understand is how they look, all right? Now, euchromatin will be lightly stained, and heterochromatin will be basophilically stained. So, what do I mean by that? Is that my heterochromatin will appear as purple and very dark. Meanwhile, my euchromatin is going to appear more hazy, more pinkish. Why? Because it's related to its structure. Euchromatin is soluble, it's dispersed. Heterochromatin is condensed. That's why the stain hits and holds onto it, all right? You can understand all of this more in your mole lecture, inshallah, okay? This is our final two pictures for the nucleus. Here, you can see the outer membrane and the individual nuclear pore complexes. You can see how porous it is, and all of these are channels that allow things to go in and outside. It allows for a communication route. And here is everything once again. So we have my DNA on the inside, my inner membrane, my perinuclear space, and then my outer membrane. Over here is my nuclear pore complex, and then my outer nuclear membrane is continuous with my endoplasmic reticulum. Why? Because both of them continue each other. When we're talking about protein synthesis, I'm gonna send the blueprint from the nucleus towards the endoplasmic reticulum. So it makes sense for it to be continuously communicating, all right? Now, before talking about the endoplasmic reticulum, let's briefly discuss ribosomes. Ribosomes are your only non-membranous bound organelle, and their main function is to assemble your amino acids. So it's the very starting point of protein making, of your transcription, tamam? And it's going to take mRNA, it's going to read it through its two subunits. So structure-wise, it's a single individual ribosome, and it's composed out of a large subunit, we call that the 70S part, and a smaller subunit, the 30S part. And you're going to discuss this in mole. So for now, we're only concerned about the structure, tamam? And sometimes I have a chain filled with, so this is all amino acids. No, sorry, this is my mRNA. And then I have a chain of ribosomes reading it. We call this chain of ribosomes polysomes or polyribosomes. Tamam? So ribosomes can exist either alone or in a chain known as a polysome or a polyribosome. Now, my endoplasmic reticulum. This is a beast of an organelle. Now, it's membranous, and this membrane is wrapped around many times, convoluted, we say, all right? And we have two kinds of endoplasmic reticulum, a smooth and a rough part. Why is that? Because we have two very different functions of my endoplasmic reticulum. My first function is protein biosynthesis, and we've been talking about this in the nucleus and the ribosomes. So it's going to take place in the rough endoplasmic reticulum. Meanwhile, I have another function known as the lipid biosynthesis. It's going to take part in my smooth endoplasmic reticulum. But when we look at the structures of both of these, why is one rough and why is one smooth? What do we mean by that? Basically, remember your small ribosomes? They attach themselves to this membranous endoplasmic reticulum. And by doing so, it gives off this feeling of being rough. So my rough endoplasmic reticulum is a convoluted membranous structure, and we have polyribosomes attached to the membrane, and it carries out protein synthesis. Meanwhile, my smooth endoplasmic reticulum, it doesn't have these ribosomes. It's lacking this extra roughness, so we call it smooth, and it carries out my lipid biosynthesis. And this is how it looks like under a electron microscope. So this is rough. You can see almost, you can actually see the ribosomes. Meanwhile, here it's a bit more smoother. You can't see any specific like bulges of ribosomes. Then, to finish our protein making journey, we have the Golgi apparatus, a different kind of organelle, which is also membranous. Now, the function of the Golgi apparatus is to modify proteins and package them. Again, you're going to talk about this in great detail in your mole lectures. So here you just need to know the function, basically. And when we're talking about the structure, it's important to note that it has two faces, a cis face and a trans face. Now, sorry, this is the trans face. No, that's correct. This is the cis face, that's the trans face. Now, you will not be asked to identify the faces. What you should know is that the cis face receives proteins, packages them, and does the work, and then the trans face is where it ships it out through vesicles. Tamam? Moving on, let's talk about the mitochondria a little bit. So, what's the function of the mitochondria? You guys know this from high school. Powerhouse of the cell, aerobic respiration, ATP production. Now, in order to carry this out, you guys will study this in great detail in mole. But for now, let's talk about the structure. The mitochondria is formed as follows. We have an outer membrane, and then we have an inner membrane that is wrapped around itself many times to increase surface area. And then we have a intermembrane space between the outer membrane and the inner membrane. And then inside the inner membrane, we have the mitochondrial matrix. How it carries out its function, we're not going to discuss that, but we should know the major structure. So outer membrane, intermembrane space, inner membrane, mitochondrial matrix. Inside the mitochondrial matrix, it's important to note that we can find traces of DNA, ribosomes, mRNA, and tRNA. And all of these are encoding for mitochondrial enzymes. All right? Now we have the weird organelles. These are organelles that are essentially either proteins or a large cell membrane bound, like a membrane bound organelle that carry out various functions. So we can start with the lysosome. And this is like a comparison slide so you can understand what each does and how it's structured. Now, a lysosome, its main function is intracellular digestion. So throughout the cell life cycle, sometimes proteins are made, we finish from them, so we need a way to get rid of these proteins. We can either use a lysosome or a proteasome. So intracellular digestion, this lysosome, if we look at the structure, it's a structure, it's a membrane-bound organelle, and inside it, we're going to have acidic, we're going to have like these lysosomal enzymes, which are stored in an acidic pH. So inside here is all the digestion happening, and all of these enzymes are very, very problematic for the cell. So it's encased inside the lysosome in a safe place. All right? If this was to be released everywhere and the cell is not maintained, it can cause cell death. Best, usually the cell is maintained and the cytosol has a pH of 7.2. So when the acidic pH inside is not met outside, these cellular enzymes will be neutralized and won't cause a problem. But for your info, you're going to have sometimes lysosomal storage disorders stemming from different structures and gene mutations. So lysosomes, intracellular digestion, that's the function. Proteasomes, similar to this, but more specific. We're going to degrade target proteins. Again, I made a protein. I'm done with that protein. I don't want it anymore, and I don't want to store it. So I'm gonna tag that protein, and then I'm gonna call out to a proteasome to come and degrade this protein. How is it structured? We have a core protein and a regulatory particle. That's all you need to know for now. Tamam? And the peroxisome is similar to lysosome, but it has a different function. This one stores oxidase enzyme oxidase enzymes. All right, and it does so. Like it interacts closely with the mitochondria, sends in enzymes in and out, and also peroxidases. So it's again like protective mechanisms and also cell cleanup. In general, you don't need to know and go into very deep details about all these three. All you need to know is their function, the structure, and specific details pointed out. All right. Also, something to note, guys, in the top right corner right here, I have the lecture which we are in at the moment. So right now we're discussing cell one, and I do this all throughout my pad. Tamam? So this is a reference point for you guys to know where we are and to know which lectures I took these information from. Hala, we finished talking about the organelles. Now we can discuss the cytoplasm. Now the cytoplasm is a very important area. You may think that the cytoplasm is only the water encasing everything. In fact, the cytoplasm is much more complicated and much more complex, not complicated. Inside the cytoplasm, we have inclusions and a cytoskeleton. Now, what do we mean by inclusions? Inclusions essentially means deposits. So we can have either lipid, glycogen, or pigmented deposits throughout the cell. Remember, the cell is a 3D space. We form a lot of these things, and they are just stored there. Tamam? So we call these cytoplasmic inclusions because they're not wrapped around by any kind of membrane. They're just existing there in the cytoplasm. And again, lipid, glycogen, pigmented deposits, all of these are found in different kinds of cells. If we're talking about a fat cell, we're going to have lipid. If we're talking about a muscle cell, I'm going to have glycogen granules. If we're talking about my skin cells, I'm going to have melanin and so on. Okay, you get the idea. And I have something called my cytoplasmic cytoskeleton. Imagine, even your small tiny cells have a cytoskeleton. And why do we need that? What do we mean by this? Essentially, I don't want my cell membrane to collapse on itself. I have organelles to push it and everything, but I need this framework to keep it up and alive and spread out. And also, it has other functions such as communication, movement, and so on. We're going to talk all about them. So we have three structures that make up the cytoskeleton. We have my microtubules, intermediate filaments, and microfilaments. All of these have specialized functions, and all of these are made out of different structures. Tamam? Let's discuss microtubules first. Microtubules, all right? So from the name, we have like this tube-like structure. Tamam? First, let's discuss the structure before the function. Now, microtubules are composed out of proteins called alpha and beta tubulin. Now, these proteins will form rows, and 13 of these rows will give me this tube-like formation. And it's going to be alternating between alpha, beta, and so on. So, at one end, it's going to terminate by beta, and at one end, it's going to terminate by alpha. The alpha row, I'm going to call that my negative end, and my beta row, I'm going to call that my positive end. These are all the details you should know about the structure. So 13 protofilaments made out of alpha and beta tubulin, and it's going to be terminated by one end on beta tubulin, and we're going to call that the positive end, and the other end is terminated by an alpha row, alpha tubulin row, and it's going to be my negative end. Tamam? What's the function of my microtubules? Vesicular transport, movement of cilia, maintaining my cell shape. Okay, you guys know exocytosis? When I take proteins from my Golgi and I put them in a vesicle, now I need to move this vesicle toward the cell membrane. Is it just going to float around my cytoplasm? No. It's going to ride on top of a microtubule and it's going to take it all the way there. Tamam? So this is vesicular transport. Also, microtubules make up cilia. If you guys know what cilia is, we're going to talk about it later. Just keep this in your mind right now. The next cytoskeleton structure is microfilaments. Hala, microfilaments, again, all of these have similar functions, but they are all different, made out of different structures. So we said that microtubules is made out of alpha and beta tubulin, and these are proteins. Microfilament is also made out of protein, but it's made out of actin. Hala, I was trying to find a way for you guys to memorize it easily, like microtubules, tubulin, microfilament. actin. Like I didn't find a good analogy, but يعني, like I tried to make it rhyme. Microfilament, actin. Maybe this horrible analogy will make you memorize. I don't know. But المهم, my microfilament is made out of actin, protein, and this actin protein has the same kind of polarity. So it has a positive end and negative end. This is not really important, but just keep it in the back of your mind. Now, it's gonna also serve the same problems. It's gonna also serve the same functions. So it's gonna also be responsible to keep the cell structure, to anchor and move certain proteins, but it's also responsible for making up microvilli. So microtubules make up cilia, microfilament make up microvilli, and we're gonna talk about this later on. Tamam? Lastly, intermediate filament. Why do we call it intermediate filament? What's this naming? What's behind this naming? Essentially, intermediate filament is in between microtubules and microfilaments. Tamam? So, if we talk about diameter, we're going to have our microtubules be large tubes, and then somewhere in the middle will come my intermediate filament, and then my smallest is my actin filament, my microfilament. Tamam? هلا intermediate filament, the interesting thing about it, it's not unified. It doesn't have a unified structure. It's not like microfilaments and microtubules. We're not talking about a single protein. We have different classes, and all of these have different places to be, different functions, and so on. But in general, its main function is to increase mechanical stability anywhere found, okay, inside the cell. So the structure, all of these different classes, their structure is made out of different proteins. Tamam? We have class 1 and 2 keratin, class 3 vimentin, class 4 neurofilaments, nestin, and so on. So it's not confined to a single type of protein. Do you need to memorize all of this? Not really. What you should know is class 1 and 2 keratin for now. Tamam? So again, just to quickly recap, microtubules made out of alpha and beta tubulin. Tamam? And also they make up cilia and they're responsible for vesicle transport and maintaining cell shape. Microfilament, actin, it's made out of actin subunits. And also it's responsible for moving stuff around and it's structure component of microvilli. And lastly, my intermediate filament is made up of different kinds of protein. It's not confined to a single kind of protein. We have different classes and its main function is to increase mechanical stability. Well, to conclude the cell, our last organelle will be my centriole. Now, centrioles are very special because they aren't permanent organelles. They appear shortly in a window before cell mitosis, all right? And their main function is they're like the microtubule organizing center for the mitotic spindle. Their main function is to ensure that the chromatids align in the center and then they're going to move them to different poles, right? So we have usually 2 in every cell. They come in pairs and they are microtubule-like structure. So they're made out of alpha and beta tubulins. Tamam? Now we can talk about the cell cycle very briefly. This is not really emphasized on in your slides, but it's important to note. I guess you guys took this from high school as well, so we're not going to spend a lot of time on it, but the cell cycle is the cycle in which the cell replicates, and also it includes the part where the cell lives. So we have G1 phase, where the cell carries out its basic metabolic functions. We have the S phase, where the cell duplicates all of its genetic information. G2 phase, where the cell is again living out its function. And then finally mitosis, which is broken down into prophase, metaphase, anaphase, and telophase, all of these consecutive phases. Over here we have a very nice diagram. And guys, this is all for your reference. Like, this is not really emphasized on, best for cohesiveness sake. We're not going to discuss this a lot. Even this isn't really a main focus for you guys, but this was mentioned, so let's briefly talk and touch upon it. We discussed cell cycle and the ability to replicate and live. What about when the time finishes for a cell? What happens? Well, cells die. They're like us. We die, cells die. And we have 2 ways cells die. It can either die by apoptosis, or it can die by necrosis. Tamam? What's the difference between them? Now, apoptosis is normal cell death, When a cell carries out its function and lives long enough, all right, it's going to come to a point where it should not replicate anymore and decide to die. So it does so by apoptosis. It's a physiological process. We call this cell suicide, and it's controlled by the BCL2 family proteins. Now, when cells don't kill themselves on their own accord, we call that necrosis. It's a pathological process. What does pathological process mean? It's abnormal. It's not the normal. So, when we have extra stresses or trauma to the cell, it can die by necrosis. Now, both of these, you guys are going to study them in very great detail in your second year. For now, it's just mentioned for cohesiveness sake. All right. And over here comes the full picture. This is everything we were discussing. This is everything. This is my entire cell. You can see the cell membrane outside, the bilayer phospholipid fluid, the phospholipid bilayer, my fluid mosaic model. You can see proteins, you can see vesicles fusing in. You can see my large nucleus right here, the inner nucleolus, my inner membrane, my perinuclear space, my pores, and then my outer membrane, and then my endoplasmic reticulum, even being continuous with my nucleus, each individual ribosome. So this is all my rough endoplasmic reticulum. My smooth endoplasmic reticulum isn't really visible. Best you can even see my Golgi apparatus, my mitochondria, inner membrane, outer membrane, matrix, and intermembrane space, lysosomes. You can see a peroxisome. You can even see centrioles. You can even see, what's it called? So this is microfilaments making up microvilli. You can see the flagellum cilia. So this is a microtubule. You can see everything. And you can even see the intermediate filaments inside, free ribosomes. You can see everything. This is the full picture. This is everything we just discussed. Throughout this PAL, we're going to have a lot of full pictures, just a moment to recap everything and reflect on what we just taken. And even actin filaments, tamam? All right, before we jump into epithelium, I think one of you asked a question. Are they like cellular reproductive organelles? Okay, I missed when you asked that question. Could you elaborate on which point? Centrioles. In a sense, if you want to think about it like that, sure. Essentially, they're responsible for mitosis, so they only appear during cell cycle. Yeah, I mean, it's not really a reproductive organelle, but it aids in it. Yeah, you can think of it like that, huh? All right, we have another question. Can you explain the difference between lysosome and a proteasome? All right, so let's go back to the comparison slide. Both of these have different functions. All right, lysosome is firstly membrane bound, and it contains lysosomal enzymes. Meanwhile, a proteasome is just a free protein roaming around, and this protein will attach to free proteins in the cytoplasm, and it's going to degrade it. Maybe they overlap a little bit in the function. Structurally, they're very different, but even in their function, they're different. Come on. Mainly lysosomes, okay, when your cell decides to die and does apoptosis, it's going to release lysosomal enzymes to the cytoplasm. That's what I was talking about. When the cell is stable, that is not a problem. But when my cell wants to die and it's unstable, it's going to rupture my lysosomes, releasing all of these lysosomal enzymes out into my cytoplasm, and it's going to tear apart everything. And the lysosomal enzymes include proteases, they include lipases, phospholipases, so they're going to break apart everything. My cell membrane, they're going to break apart my proteins, my carbohydrates, everything. Tamam? Meanwhile, proteasomes, they only target free proteins. Tamam? I hope that answered your question. And technically, proteasomes are not membrane bound, okay? All right. Do you guys have any more questions before we move on to epithelium? So we just finished 2 lectures. We have 4 more, and inshallah, it's going to be very smooth sailing. No worries. Yeah, let's begin. Now we're going to discuss epithelium. You guys are finally studying histology. We finished the baby stuff. We finished the cell, the foundation, all right? Now we're going to study histology. But before that, you guys should understand some basic principles in histology. So, Histology, again, we said it's the study of tissues, right? It's the study of tissues under a microscope. Now, in order to prepare a sample tissue, we have something called tissue processing. And you should know the order of it. And you should also know each of these stages, the definitions of them. So we're going to start off with fixation. We're going to preserve the cell and the tissue structure. We're going to dehydrate it, remove all the water from it, and replace it by alcohol. Then we're going to clear out the alcohol, and we're going to add paraffin. Paraffin. And then we're going to make sure that the paraffin is infiltrated and it's spread out as much as possible. And this is happening at 52 degrees to 60 degrees Celsius. And then my last and final step is my embedding step, where I'm going to make this melted paraffin relax, allow it to harden, and I'm going to have a sample of my tissue. I'm going to take it into a microtome, which is going to slice it very thin and present me with a slide. Now that I have a slide, I can look at it under a microscope. But in order to make sense of what I'm looking at, I'm going to stain it. Why do we stain in histology? Because staining will differentiate things from things. Essentially, your organelles and the cell structures, all of them have different properties and all of them interact differently with stains. So, one of the most used stains in histology is hematoxylin and eosin, H&E preparation. This is done for all slides in histology. Tamam? Hematoxylin will bind tightly to my DNA. Eosin will bind tightly to my cytoplasmic structures. So I'm going to have two different differentiations, two different colors, like a color pattern. My DNA is going to be stained purple or blue, and my cytoplasmic organelles is going to be stained pinkish. Halla, under a light microscope, can I see a mitochondria or my rough endoplasmic reticulum? Can I see a ribosome? Not really. These things are even smaller than light itself. We need something smaller than light. We need electrons to be able to process them. So when I apply my stain, I'm going to have two different regions. I'm going to have, firstly, you can see the cell membrane. You can see the borders. But inside the cell, I'm going to have two regions. I'm going to have my nucleus and my cytoplasm. Again, with the compartments. Tamam? So hematoxin is going to attach to DNA, staining it purple or blue. And eosin is going to stain my cytoplasm pink. All right. All of this, you guys should know the steps. You should know the order. You should know the definitions. They could ask you on MCQ. You don't really need to know what a microtome is, but you should know what stain is what's the color of the stain and what the stain does to the cell. Tamam? Sorry. Now, finally, we can talk histology. We can discuss types of tissues. طيب, what is tissue? Again, let's talk about the definition of tissues. Tissues is an assemblage of similarly specialized cells united in performing a specific function, right? So we're assembling a group of cells that are similar to do a specific function. It's made out of extracellular matrix and these specialized cells. And in tissues, we only have four types. We have epithelium, connective, muscular, and nervous. And all of these are made out of different kinds of cells and different ratios of extracellular matrix. Okay? We're going to first discuss epithelium, and then we're going to discuss connective tissue. But this pal will finish before us discussing muscular and nervous. This will be covered in another pal, inshallah. Tamam? So let's jump straight into epithelium. Again, we should know function and we should know structure. Okay, tissue, we said it's a group of cells carrying out a function. What is the function of my epithelium? My epithelium is separating my internal environment from my external environment. So inside my body, I'm going to have different cavities, I'm going to have different structures, and I want to separate my internal environment from my external environment. And also, besides the covering function, I have absorption, secretion, excretion, gas exchange. Well, how? Okay. Habbe. We'll talk about everything, all right? Let's talk a little bit about the structure of epithelium, and we're going to discuss it into great detail. The epithelium is going to be a sheet of cells that will line my body surfaces, my inner and outer body surfaces. Epithelium covers everything in my body, everything from the inside and the outside, except for three places. Except for my articular cartilage, my teeth enamel, and the anterior surface of my iris. So anywhere I have a joint, my articular cartilage doesn't have epithelium. Tamam? Should you know the exceptions, just keep them at the back of your mind. Know that it covers everything. Tamam? It doesn't have a blood supply, so nutrients are delivered by diffusion. It has little extracellular matrix, renewed by mitotic activity, anchored to a basement membrane structure. What is all this? Okay, we're gonna talk about everything. Yalla, bismillah. Starting with the basement membrane, the idea of a basement membrane. Halla, if I'm building a tissue, all right, I'm building a tissue. I have a group of cells that I want to layer on top of each other. Now, in order to make it more oriented and organized, I'm going to have a basement layer, a basement membrane, and on top of it, I'm going to build my cells. That way, it's not messy. That's way it's organized. That's way not everything is going in and out. So my basement membrane is a layer most underneath my epithelium. Tamam? It can be facing up, it can be facing down. Every epithelium starts with my basement membrane, and then we build on top of it. So the function of it, semi-permeable filter for substances, but it's to build epithelium on top. Tamam? How is the structure of my basement membrane? Now we're going to talk. This is very important. My basement membrane has two layers. It has a basal lamina and a reticular lamina. My basal lamina is a thin layer of fine fibrils, and it's composed out of type 4 collagen, protein called laminin, proteins called nidogene and perlecan. Tamam? My reticular lamina is more diffuse and fibrous, and it's made out of type 3 collagen. What do I mean? What am I talking about? Let's discuss this. Hello. All right. When we're talking about the basement membrane, we should understand two things. This basement membrane is composed out of two layers, a basal layer and a reticular layer, and these layers should be attached to themselves very tightly and should attach themselves to the epithelium very tightly. Tamam? Because I don't want my basement membrane to be flimsy. I want it to hold everything in place. Halla. I'm gonna look at my basal lamina, how it's formed, and then how my reticular lamina is formed. Again, my basal lamina, type 4 collagen, laminin, nidogene, perlecan. You should know all of these proteins, tamam? Halla, this is how everything looks like. This is an epithelium cell, and this is my basement membrane, but it's opened up. Both of these layers are my basal lamina. We're gonna have a layer in the basal lamina made out of laminin protein. And we're gonna have another layer made out of type 4 collagen. Remember laminin and type 4 collagen? So we have a layer of laminin and a layer of collagen. This is still the basal lamina. We're not talking about the reticular, come on? But between them, I need some way to connect them. What's going to connect them is my nidogene and my perlecan proteins. So I have two layers. This is a layer of proteins. This is another layer of proteins. And in between them, I'm going to have proteins connecting them. It's hard to see, but it's better on this diagram. So if we zoom out, laminin is bound to type 4 collagen through nidogene and perlecan. So nidogene and perlecan, we can say cross-link laminin and type 4 collagen. طيب, I connected both of these layers. How am I going to connect it to my epithelium? We have a protein in the epithelium called integrin, and we have a protein, laminin, we just talked about that, in my basal lamina. Both of these proteins are going to interact with each other, are going to connect, cross-link. So, integrins is found in the epithelium cell. Laminin is found in one basal lamina layer. Both of these are gonna form crosslinks. All right, my integrin now is bound to laminin. This is my first layer. So underneath that, I'm gonna have another layer, which is my type 4 collagen, and that layer is bound to laminin through nidogene and perlecan. All of these proteins you should understand. All of these proteins you should know and memorize their names. Tamam? Most thing you should associate basal lamina is type 4 collagen. Tamam. That was the basal lamina. What about the reticular lamina? And we're going to recap all of this. Don't worry. Reticular lamina is made out of a layer of type 3 collagen. Like, I'm talking about collagen this, collagen that, type 4, 3, 7. What's all this? We're going to talk about it later. For now, just keep in mind, we have different kinds of collagen. We have collagen type 4, type 3, and each of these are found in a different layer. هلا, collagen type 4 is in my basal lamina. Collagen type 3 is in my reticular lamina. هلا, my reticular lamina is going to be a layer underneath my basal lamina. So I'm going to have to connect these two layers together. I don't want it to be flimsy. In order to do that, my type 3 collagen reticular layer and my basal lamina, I'm going to cross-link it through type 7 collagen. All right? In a sense, I'm connecting type 4 collagen to type 3 collagen using type 7 collagen. It's so confusing, so don't confuse things. Keep drawing it out, keep saying it multiple times, but let's have a look at this structure right here. Again, basement membrane, epithelium. I want two layers of my basement membrane to hold tightly, and this layer to hold tightly to my epithelium. My epithelium will have integrin receptors, my integrin protein, and this is a protein found in the cell membrane. This is the cell membrane of my epithelium. Remember when we talked about the cell membrane? It can have proteins. Integrin is one of these proteins. Now, this protein will interact with the layer of laminin in my basal lamina. And then it's going to cross-link integrin to laminin. So, epithelium to basal lamina, good. Now, basal lamina to basal lamina, how do we do that? We have a layer of laminin and another layer of type 4 collagen, and we cross-link it through perlecan and nidogen. Then we're going to take a look at my reticular basal membrane. You see how my basal membrane, basal lamina here is very thick and connected? My reticular is more so fibrous, less dense. Here is my reticular basal membrane, and here's my basal basal membrane. My reticular is made out of type 3 collagen. My basal membrane is made out of type 4, and it's made out of laminin and integrin binding. صح؟ هلا, type 4 collagen will bind to type 3 collagen using type 7 collagen. Don't confuse this, guys. I know if you want me to repeat it, I'll repeat it later on, but we need to move on from basal membrane, تمام? هلا, we said that we have a basal membrane, and on top of it, I'm going to build my epithelium, right? طيب, now that I have a reference point in a sense, I can kind of orient myself towards the cell. We have this cell, my epithelium cell. We have the area that is close to the basal membrane, and we have the area that is farther from the basal membrane. So I can call this the basal surface. I can call this the apical surface. And it's very important. Why? The apical surface away from the basal membrane has specialization. We're going to talk about it in great detail. And also, I have a lateral domain. And now we discussed that in my basal surface and my basement membrane, I want it to connect tightly. Like, what about my cells when they're next to each other? Do I want my cells to be flimsy, not be connected and held tightly? No. I need this lateral domain to hold the cells in row together. So, you see how these cells are in row? I want this lateral domain to hold everything together. So we have a lateral domain, apical domain, and a basal domain. تمام? And all of these have different functions. We're going to discuss it later. Let's talk now about epithelium. هلا, epithelium is a type of tissue. We said that it has a lot of different functions. We said secretion, we said absorption, gas exchange. Those are very different functions. How does that happen? How does that work? Essentially, to meet these functions, epithelium comes in different shapes. So, when we classify epithelium, we should classify it based on its shape, then the arrangement, then any apical specialization add-ons. طيب, classification. If we talk about the shape, we have three shapes that epithelium can come in. Squamous, which means flat, cuboidal, cuboid-like, and columnar, column-like. And then when I'm looking at the arrangement, I can have a single layer of epithelium, or I can have multiple layers of cells on top of each other. We call that stratified. And I have also something called pseudo-stratified, something called transitional. We're going to talk about that in a minute. The epithelium, is it made up of the basement and the reticular one, or is it a separate layer? All right. هلا, basically, the epithelial cell is its own thing. It's interacting with the basement membrane. But when you're talking about epithelial tissue, you include the basement membrane. The basement membrane forms the first step, and then on top of it, you're going to have the cells. Okay, did that answer your question? Hello. Here we have a comparison slides, and these are our different kinds of epithelium. Simple squamous epithelium, simple cuboidal and columnar, and then we have something called pseudostratified columnar epithelium. All of these look very different, and they look very different because they have different functions. If I'm talking about simple squamous, which is the very flat nucleus, very flat cells, very small, narrow cells. Okay, thank you, Tamam. These cells, they minimize the distance between the apical and the basal sides. Why would I need to minimize the distance? Because I need things to be moving up and down. So it will allow for diffusion of molecules and filtration of stuff. Where do I need this diffusion? Where do I need this filtration? In capillaries, in the lungs, and in kidneys. In lungs, I need to get in oxygen and I need to push out carbon dioxide. So my epithelium that will line the innermost surface of my lungs, it's going to be simple squamous epithelium because of its small distance and it allows for rapid diffusion. Same story for the kidneys, tamam? هلا, columnar and cuboidal epithelium, they look very different and they have a larger number of mitochondria because they have a very different purpose. They serve a completely different function. Their function is the secretion and the absorption. All right? So we said that the functions of epithelium are what? Covering everything. That's covered by all types of epithelium. But then we have diffusion. So that is covered by simple squamous. Absorption and secretion. This is now getting covered by cuboidal and columnar. So using the mitochondria, we're going to have ATP and it's going to be used in secretion and absorption. Now we have a specific kind of epithelium called glandular epithelium, and that is the secretion kind of epithelium. Absorption is found in the intestines and also in the kidneys. Tamam? For your level, what you should know is the function of epithelium, how it looks like under a microscope, and also where you could possibly find it. Tamam? Pseudostratified columnar epithelium. This is a tricky one. Stratified, we said, is layers, multiple layers of cells. When I'm talking pseudo means fake layering. What am I talking about here? Let's look here. This is my basement membrane, and on top of it is my epithelium, right? So if I have one layer of cells, all of them connected to my basement membrane, it's called simple epithelium. Over here, I have cells. They appear to be on top of each other, so I may think it's stratified, but in fact, all of them are connected to my basement membrane. All of them. So they are giving the illusion of stratification, but they're all on the same level. So even though the nuclei are not aligned on the same row, even though they're on top of each other, it's one layer. And we call this pseudostratified. This is how it looks like. This is the basement membrane. You can see each circle here is a nucleus, and you might think they're on top of each other, but in fact, no. It's hard to do, but if you trace them all, it's all on the same membrane. So we have a question. What kind of epithelium does the skin have? Amazing question. We'll get shortly there, all right? Hello. It's important to note the structure of pseudostratified epithelium, where it's staggered appearance of nuclei, but it's true simple because all of them rest on the prominent basement membrane. And its function is ciliary movement. Remember when we talked about cilia earlier on? Cilia is a kind of apical specialization. We're going to talk about it, but it's responsible to move things. And we can see cilia right here. So this is specifically found in the lungs, nowhere else. All right? Now we can talk about stratified epithelium. So we have stratified squamous epithelium and we have transitional epithelium. And the stratified squamous epithelium is the kind of epithelium on the skin. All right, so that's to answer your question. It's stratified. We have more than one layer because we need the extra protection. And it's squamous. Why? Even though it's kind of tricky to see the cells over here, we're going to look at the topmost layer and we're going to see flat cells. So I'm going to say it's stratified due to the many layers of cells. and it's squamous because of the topmost layer. I will not look over here, I will only look at the top layer, tamam? So, stratified, squamous, the superficial cells are squamous. What's the function? Protection, waterproofing. Location, the skin. تمام؟ هلا transitional epithelium is the same story in the sense that it's multiple layers and it serves the function of protection, but it is specific and unique due to its structure. In transitional epithelium, first we call it transitional because we have different sizes of cells. We have small cells becoming bigger, smaller, and then bigger again. And we have characteristic umbrella cells at the top. Can you guys see the umbrella? تمام? So we have umbrella cells at the top, and we have different kinds of cells, different shapes of cells all throughout. So we call this transitional epithelium, and it's only found in the urinary tract. So my urethra and my bladder and other parts of the urinary tract. So we can call it urothelium. All right. And its main function is to accommodate the stretching that occurs when fluid fills up my urinary tract. So when I'm making urine, I'm going to compress all of these cells on each other to allow for the stretching of my bladder. And when the urine is released, my cells will open back up. Tamam? And also, these cells can withstand the toxicity of urine. Tamam? So, what kind of epithelium does the skin have? How can the squamous secrete sweat? All right, we'll get there. MashaAllah, nice question. So, we're on the right track. We'll get there. All right, we talked about the simple epithelium and we talked about the stratified epitheliums. Now, let's talk about the epithelial add-ons. Halla, if you guys remember, when we talk about the polarity, the apical surface and the basal surface, these add-ons are on the apical surface. All right, they're away from the basal membrane. And we have around 4 types. We're going to discuss these ones for now. We have cilia, keratin, microvilli. Halla, cilia is responsible to move things around. Microvilli is responsible for absorption. You know what villi is? The thing in your intestine? The villi is the big thing, and it's made out of small, tiny microvilli, and all of these are for absorption. And then we have keratin, which is a layer of just protein, and it's just adding to the layer of protection that my skin has. It's only found in the skin, and like when we talk about epithelium, it's only found on my skin. Tamam? Again, what is keratin, guys? We just talked about it like 20 slides ago. Keratin is an intermediate filament, class 1 and 2. That's why I highlighted it back then, because I want you to know it. Tamam? So it's an intermediate filament, but over here it's a protein serving as extra protection on top of my stratified squamous epithelium. So this is the epithelium of my skin, and all of this is extra keratin. Like, is this everywhere? Is this all around my skin? No. Keratin is only in special places where my skin will be thicker. And it's my palms and the soles of your feet. Halla, this may have a little bit of keratin, but not to this degree. This is the difference. The skin of your palms and soles have a large layer of keratin. Tamam? Halla, let's talk about microvilli. Let's talk about cilia and all of them. Microvilli, tamam, we said that its main function is for absorption. It's going to increase surface area for absorption. And if you guys remember, when we were discussing microfilaments, we talked that it makes up microvilli. So microvilli is made out of actin filaments, and they are finger-like cytoplasmic projections outside the apical surface of my epithelium. Let's imagine this. So where am I going to see microvilli? I'm going to see it in my intestine. I'm going to see it in my absorption. Which kind of epithelium carries out absorption? Columnar or cuboidal? صح? So, on the columnar and cuboidal epithelium, Over here at the top, I'm going to have microvilli on my apical surface. Okay? And its main function is to increase surface area for absorption. You can see it again here. So this picture is a bit tricky. Basically, the basement membrane is running HEG, and this is the cell. So this is the apical surface, and this small hazy pink line is actually microvilli. These are simple columnar epithelium. Tamam? This is everything you should know for microvilli. So it's again microfilaments, actin filaments, and it mainly carries out absorption. It's finger-like projections. Cilia, on the other hand, is hair-like projections, and you can clearly see and identify the hairs on top of the respiratory simple columnar epithelium, or sorry, pseudostratified epithelium. هلا, these hair-like structures have like a basal body, so it's similar to like a secondary basement membrane. We call it a basal body, and essentially, the cilia is not made out of what's it called microfilament. It's made out of microtubules, so alpha and beta tubulins. It's arranged in a special way. It's not arranged in a 13 protofilament thingamajig. It's arranged in a 9 what's it called, like a 9 doublet right here, a 9 doublet microtubule, and then in the center we're going to have 2 central microtubules, and we call this an axoneme. Tamam? Nexins, this is a protein, will link the peripheral doublets to other proteins. Tamam? What you should really know is where it's found and its function. So cilia, highly motile, hair-like processes that will move things around. Where do I find it? In my respiratory tract mainly, so over pseudostratified epithelium. Tamam? We have a question. The first picture, is this the correct question? 1 second. The first picture is transitional or pseudostratified. Can you tell me which picture you mean? This one? All right. No, this picture is not pseudostratified. Or sorry, this picture is not transitional epithelium. Let me try and do something. If we look at the bigger picture, all right. Oops. Here's the bigger picture. This is pseudostratified epithelium. It's very messy. I don't want to scare you guys, but this is the basement membrane, and all of these cells are confined to one. Tamam? What I wanted you to focus on is the hair-like processes, which we call cilia. That's all I want you guys to focus on. Tamam? All good? Let me show you transitional epithelium one more time. Transitional epithelium is true stratified, okay? These cells are not all touching the basement membrane. They're very different. And we have umbrella cells. You couldn't see umbrella cells you couldn't see umbrella cells over here, tamam? These are not umbrella cells. And don't worry, the slides that will come in your exam, they're not as scary. Inshallah, they're going to be very easy and straightforward. Don't worry. Like in OSPI, they can ask you about histology, in MCQ, they can ask you, and SAQ, they can ask you, but inshallah, the slides that will come, they're always clear because they're not trying to be that mean, at least for now. All right, you never know. Hello, stereocilia. Stereocilia, let's see, did that answer your question? Tamam. All right, stereocilia is similar to cilia, but not exactly. Stereo means similar or mimicking. So it's similar to cilia, but it's actually functioning as microvilli. So the function of stereocilia, facilitating absorption. It's not moving things around. And we only find this ever in the male reproductive system. So it's very low yield. It's just something to take note of now. But later on, when you study endorepro, it's going to be a whole topic, okay? Just keep it in mind. It's another apical specialization, and it's called stereocilia. Now, let's take a look at the full picture. Let's appreciate what we just studied, all right? So, epithelium. We said we look at the shape of it first, and then we look if it's layered up or not. This is simple squamous because it's squamous cells and it's one layer. This is cuboidal and it's simple because it's one layer over the basement membrane. This is columnar, this is pseudostratified because all of them are touching the basement membrane. And then when we're looking at stratified, we have stratified squamous. We're going to look at the topmost layer for the squamous cells. We have stratified cuboidal, stratified columnar. You don't really need to take note of these for now. Transitional. We can see clearly larger umbrella cells covering multiple cells underneath them. And also we talked about microvilli. We also, do I have another cell? All right. We also talked about microvilli. We also talked about cilia, stereocilia, and all of these. All right, guys? Let's practice really quickly. Yalla, guys. Can someone tell me in the chat what kind of epithelium this is? I want you to give me the full name. Yalla, just a bit of interaction, and then we're gonna solve 3 together, and then I'm gonna solve the rest of myself, so we don't take a lot of your time. Simple cuboidal, perfect. This right here. I know the image is blurry, but yalla, guys. This is the basement membrane. Each single cell here is whoa. MashaAllah, everyone is okay, everyone is replying. It's amazing. Alhamdulillah, okay. So stratified squamous. MashaAllah, okay, perfect. This is again stratified squamous. Yalla, this one, guys. What do you think this is? Stratified squamous keratinized. All right, that's a good guess for this one. But for now, let's look at this. What do you guys think this is? It's the one that's hard to type. Aywa. Good. You guys guessed it, I believe. Pseudostratified ciliated. Like, what's this epithelium here? You see this? What's this epithelium here? Let's hear it. Yeah. The keratinized. I got what you meant. Don't worry. Simple squamous. Perfect. What do you guys think this is? These are long, elongated cells. These are not cuboidal. So simple columnar. Perfect. And this, ignore this. What do you guys think this is? Yes, simple columnar for the one before this, correct. This is the basement membrane. And then we have all of these cells, and then we have a large keratinized. Okay, someone said simple squamous. It's stratified squamous, all right? Let's not mix that up. Because we have a lot of cells here, tamam? So it's stratified squamous, keratinized, correct. Okay, mashallah, you guys are really getting everything, salah. All right, what do you guys think these small projections are? So this is pseudostratified. This is cilia. Thank you. Can you explain? Okay, this is not microvilli. This is cilia. Microvilli is very smaller than cilia. You can't really see it properly. You can't see like individual hairs popping out, right? So this is cilia. This is not microvilli. If I want to show you microvilli, you can think of it and see it just as like like a hazy pinkish thing on top of the apical surface. All right, that is microvilli. Don't worry, the pictures inshallah they will be so it's almost not visible. Yeah, it's like a hazy thing. But you can point it out. Yeah, and why it's not visible, it's visible at the same time. Why? Because when you don't have it, the apical surface is clear. It's white. There's no this pinkish haze. But when you have this pinkish haze, you have microvilli. All right. And which type of epithelium is stereocilia? هلا stereocilia يعني it's again honestly I'm not sure. I'm worried it's either pseudostratified, but from my knowledge, pseudostratified is only in the lungs. Yeah, so can you fact check this? All right. So I don't give you guys any wrong information. But you don't really need to bother yourself a lot with stereocilia right now. تمام. هلا let's talk now about the lateral domains. We talked about my basement membrane and we talked about my apical surface. صح? Let's briefly talk now about my lateral domains. All right, we have some questions. Okay, thank you. تمام. هلا, what's the purpose of my lateral domain? I want to hold on to my neighboring cell as tight as possible. I don't want it to go anywhere, all right? And we do it by three different kinds of junctions. We have something called tight junctions, anchoring junctions, and gap junctions. Okay, so thank you, Yasser. We have the facts now. Stereocilia will be seen on pseudostratified epithelium found in the male reproductive tract. Tamam? Don't bother yourself with this. Inshallah, it's not gonna be tested on. This is something completely for EndoRepro later. Tamam? هلا هون, let's stay on topic. We have three kinds of junctions that connect my cells to each other. هلا I'm saying three kinds, but we have two exceptions. We have tight junctions connecting cell to cell. We have gap junctions also connecting cell to cell. But when I'm talking about anchoring junctions, only my adherents and my desmosomes connect cell to cell. My hemidesmosomes and my focal adhesions connect my cell to my basement membrane. You're gonna see how we foreshadowed this, by the way. Okay, you'll see how. Mashi, let's talk first about tight junctions. And all of these, we have a lot of proteins. You guys should memorize and know the proteins properly, all right? Halla, tight junctions, it's my most apical component. What am I saying? Apical, we're on the lateral domain, right? Yes, we're talking lateral domain. Yes, we're not talking about the apical area. Halla, anything away from the basement membrane, we can call it apical regardless. So I'm talking in regards to my lateral domain, the most top one, the most apical one in a sense. So it's my tight junction. You see how it's on top of everything, but it's my lateral domain. It's not my apical domain. تمام? هلا, my tight junction, it's gonna be, the main function of it is to hold the cell tightly. تمام? It forms a seal. It's not continuous. It forms multiple seals, but it's gonna connect and hold the cells tightly. We call it tight junction because of the large amount of proteins involved. How many proteins do we see here? Occludins, claudins, junctional adhesion molecule, we can call it JAM, and ZO1, ZO2, ZO3. So we have 1, 2, 3, 4, 5, 6 proteins involved in this. It's very tight, it's very strong. Tamam? Halla, claudins and occludins, and I want you guys to know their names and know which is for what. So claudins and occludins is for tight junctions. I don't want you mixing them up. Mashi? They have four domains, one domain. These details not really matter, but what matters is that over here I have a an occludin molecule, and it's connecting both sides Hic. My claudin is connecting both sides Hic. So my claudin to claudin interact. My occludin to occludin interact. My jam to jam interact. All right, so this is the jam of cell number one. This is the jam of cell number two. And they are the ones interacting with each other. We're not talking two different proteins. They're the same protein interacting, but from different cells. This is not a laminin integrin situation. All right? Halla. The ZO1, ZO2, ZO3, they are responsible to, they all interact with occludin. Tamam? They interact with everything. But you should take note of ZO1 specifically, because ZO1 will then further attach itself to the actin microfilaments of my cytoskeleton. Tamam? So my cytoskeleton microfilament is attaching itself to my ZO1 big olive right here. I call them olives because that's what they look like. Tamam? So, tight junctions, occludins, claudins, jam, ZO1, ZO2, ZO3, my olives. And my olive number one, ZO1, is connecting itself to actin. Tamam? Jam, occludin, claudin, and then these things are attached to the ZOs, and ZO1 is then attached to my microfilaments. And this is for your info, just read it on your own. Hella, we finished tight junctions. You saw the amount of proteins involved. That's why it's very strong. That's why we call it tight junction. Now, if we're talking about the adherence junction, we have one protein, cadherins. All right? So cadherins, it also serves the same function, and it's gonna hold the cells tightly together. Cadherin will bind another protein called catenin on the inner surfaces of my epithelium. And then catenin, by one way or another, will attach itself again to actin. filaments, so microfilaments. All right, we have a question. Do we need to know their order, like which is on top? Do you mean like tight junctions first and then adherence junctions, or do you mean jam, occludin, claudin? And the jam, occludin, claudin, not really. But you should know and know. Yeah, you don't really need to know which is on top. No, just know that they're responsible for tight junctions. And they could tell you in an MCQ, oh, so a person has a genetic mutation there, they have faulty tight junctions. Which of the following proteins is affected? All right, they can put one of these, and then they could put, for example, cadherins. All right, I'm not gonna choose cadherins. They're not responsible for tight junctions. I'm gonna choose one of these, the one they put for me. So that's why I'm telling you to know them. All right, and to know which is most apical. So tight junctions comes first. Then under it is my adherence junction. My adherence junction is not as strong. It's one protein, cadherin, and it attaches itself to catenins and then actin filaments. All right. This is the mechanism, but you don't really need to focus on it. Halla. These are anchoring junctions. In my anchoring junctions, I have two that attach cell to cell, my adherence junction and my desmosome, and I have two that attach cell to basement membrane. Cell to basement membrane. Tamam? Let's finish first the cell to cell. We finished adherence junctions. Let's talk about desmosomes. Desmosomes are the easiest thing on the planet. They are two proteins, desmoglein, desmocolin, from the name desmo, easy peasy. All right. Function, same idea. We're connecting these cells together, but we're not attaching ourselves to actin. We're attaching ourselves to intermediate filaments. Do I care what kind of intermediate filament this is? Could be GFAP, it could be keratin. I don't care. Fibronectin, I don't care. All I need to know, desmosomes is made out of desmogleins, desmocolins. It attaches to intermediate filament. Okay? Now, let's talk about my cell to basement membrane. We have hemidesmosomes, and we have focal adhesions. Hemidesmosomes, we talked about it indirectly. You guys, we didn't name it. We didn't name drop it back then. But remember integrins, which are parts of my cell membrane, the proteins in the basal cell membrane? Integrins, that integrin complex was part of my hemidesmosome. So my hemidesmosome will attach my epithelium to my basal membrane, my basal lamina. That thing that we talked about, integrin and laminin, which attach itself cross-links normally, it does that through a hemidesmosome. هلا we also have collagen type 17 involved and plaque proteins. Don't get into the details. All I want you to know is what makes up hemidesmosomes. Integrins, type 17 collagen, and these plaque proteins. If you got to memorize them, good for you. But they get the picture and ask us what type of junction it is. Will they do that? I don't think so. I mean, I remember from our exams, it's never that hard. It's always like, oh, this person has a problem with something. What's the protein defected? Okay. So that's why I'm telling you guys to memorize the proteins. Desmoglines, desmocolins, the word desmo, it's a clear giveaway. Cadherins for adherent junctions, the occludins, claudins, junction for tight junctions, and so on. Tamam? And desmosomes, integrins, type 17 collagen. And I know this is like the fifth collagen. We're going to discuss collagen, don't worry. And then we finish hemidesmosomes. Lastly, on our anchoring junctions, our focal adhesions. These are complicated, these are confusing, so just know that we have, like the way they're structured is broken down into three areas. We have a cytoplasmic phase, a transmembrane phase, and an extracellular phase. All right? هلا, the transmembrane phase is my integrin again. All right, so the same integrin, my hemidesmosome, is also sometimes involved in my focal adhesion. My integrin is going to attach to fibronectin, which is my extracellular phase, and all of this is my focal adhesion. And then my cytoplasmic phase has like 4 different proteins, talin, vancomycin. You don't need, inshallah, you don't need to know that. All right, it's a headache to memorize all of this, and it's so much energy. The function to link the actin cytoskeleton to these extracellular proteins and to the basal lamina. So it's a way to anchor everything the entire cytoskeleton to the basement membrane. I'm anchoring it here to the cell, to the basement membrane. تمام. Finally, gap junctions. This is the easiest one. This is the nicest one. This is the greatest one. I love this one. Gap junctions. All right. Their function to me called connexin protein. 6 of connexin will form a connexon. All good? Can you guys see my screen? Like again, the full picture. One last time, I guess. So adherent junction, desmosome, hemidesmosome, and down here should be also my, I forgot what's it called, focal adhesion, sorry, and gap junctions. Tamam. And now we can talk about glandular epithelium. If I recall correctly, one of you guys asked on the chat, how can a sweat gland secrete or something? It does so by glandular epithelium. So if you guys remember, we said epithelium has the function of absorption, covering, diffusion, secretion. How does it secrete? It's a specialized form called glandular epithelium. We have two kinds of, you have exocrine glands and endocrine glands. And what happens is, when you are in your mom's womb, when you're an embryo, epithelium will invaginate and push down, infiltrating. Yes, sir. Is the internet good or what? All right. Sorry, guys. It's going to infiltrate the underlying tissue. As it does so, it's going to form this gland-like structure. By doing this gland-like structure, I'm going to have a duct. I'm going to have a form of communication. Sometimes this form is preserved. Sometimes it's cut. If it's cut, this gland will still does its secretory function, but it doesn't have anywhere to push its secretions out. So blood vessels will wrap around it and it's going to secrete directly into the blood. So we're going to call these glands, with the connection maintained, exocrine. They have a duct. And the glands that have lost this, they don't have a duct, endocrine. So endocrine, that's an entire block later on, and it's mainly responsible for the secretion of hormones. And for your info, the pancreas is the only organ in your body that has both kinds of blood. Exocrine and endocrine, this is just a fun fact. You're going to take this in GITI. When we're talking about the structure of these exocrine glands, two main places, or like, let's say parts. First is the secretory portion, and then obviously the excretory duct, because we're talking about exocrine glands. And now the secretory portion can either be a single cell, one cell with its duct, or one to multiple cells, 1, 2, 3, 4, 5 multiple cells, all secreting into the same duct, and that is pushing my secretions, right? So we're gonna call my goblet cell here a unicellular duct, and my complex sweat gland here a multicellular duct. Now we call these cells acini, and the entire gland is adeno. If you hear about the word adenopathy, adenoma, we're talking about glands. Any specific glands? No, generally, just any form of gland, whether it's a sweat gland, mucus gland, anything. The function is secretion of products that are used in another location. I'm not using these products at the secretory portion. No, I'm secreting this elsewhere. For example, in my stomach, I'm secreting hydrochloric acid. I don't want hydrochloric acid in the gland itself. I want it in the stomach. So through the duct, it's going to go in. Now, the mechanism of secretion is as follows. It can be either merocrine, apocrine, or holocrine. What do I mean by that? Comparison slide. All right. These are the three different mechanisms of secretion. Most commonly, merocrine, which is essentially exocytosis. I believe you guys are familiar with the term, but if you're not, it's basically pushing out the secretions through membrane-bound vesicles. They will fuse with the cell membrane vesicles, and it will push it out. So this epithelial secretory part will push out into the duct through merocrine secretion. And this is common everywhere. Let's take, for example, pancreatic acidosis. Apocrine and holocrine are very different mechanisms, but they only happen in certain areas. Apocrine secretion occurs in a lactating mammary gland, where instead of releasing the vesicles and not losing any kind of cell membrane, we're going to pinch off the cell membrane ever so slowly to release a little dropules into my duct. So for an lactating mammary gland for pregnant ladies, whenever they want to make the milk, this is their mechanism of secretion, apocrine secretion. Now, holocrine secretion is very dramatic. It's very suicidal. I don't like it. It happens in the eyelids. So you have a gland over here responsible for your eyelids. What do we call it? Ears. Sorry, I forgot that word. So in order for that secretion to occur, it's through the tarsal Mobian gland and it does it by holocrine secretion. So essentially, it's like a virus infection where the secretions increase, and accumulate inside the cell until the cell bursts out, releasing all its contents and the secretions into the duct. Well, with this, we finish. Do you guys have any questions before moving on to connective? Let's hear some questions. Could you please repeat the gap junctions before preceding? Sure thing. So you want the gap junctions, and I believe someone else wanted. Can you talk about the difference between focal and adhesions and hemidesmos? All right. Hello. Gap junctions. They are lateral connections, cell to cell, but these connections aren't to hold these cells together. It's a gap between the junction, and it's just a channel allowing things to move in and out across the cells. All right. The way it does it is through a protein subunit called connexin. 6 of these will form a connexon-like gap junction, which will then communicate both cells together. So it's just a hole between these two cells. It's just a tube between these two cells. All right? That is our gap junction. So someone was asking hemidesmosomes and focal adhesions. If I can explain them again. Hemidesmosomes and focal adhesions, both of them serve the same function. We want to attach my epithelial cell to my basement membrane. Hemidesmosome does it the way we discussed it earlier when we were talking about integrins and laminins. Remember in your basal lamina, the laminin layer? You had the laminin layer, and then my integrin protein connecting itself and cross-linking with my laminin layer. And then underneath it, I have my type 4 collagen, which was connected by, what was it called? Protein, I think, I forgot, but you guys should not forget in the exam. But essentially, this is how my basal membrane is. For my hemidesmosome, it's the same story. Add on top of it plaque proteins and type 17 collagen. All right. Meanwhile, focal adhesions serve kind of a different function. That one, we are connecting also like intermediate filaments to my basement membrane. Here, I'm going to connect my actin microfilaments. So both of them are also connecting my cytoskeleton to my basement membrane because I want my cell, it's like a hot, I don't know how to describe it. It's like, let's say, it's like a kite. Like my kite is going up. I need to hold it and stamp it somewhere. Like connect it somewhere. So I'm going to do that. I'm going to connect it to proteins, and then these proteins on my cytoplasmic face will connect to my integral protein on my transmembrane face, that then connects to fibronectin, that then connects to vasoactin. It's a long road, but it's important to know. Right? You're welcome. Do you have another question? No questions. All right. So for our last segment, which is connective tissue, I think this is taking a little longer than I wanted. So inshallah, we're gonna fly through connective tissue. It's very simple. So guys, we finished the first kind of epithelium. Sorry, the first kind of tissue. You guys studied epithelium. Now you guys know all the ins and outs about epithelium. Allah. You guys linked structure to function. Now we're going to do the same story, but we're going to discuss a different type of tissue. We're going to discuss connective tissue. Connective tissue. What's the function of it? It's the major structural constituents of the body. And it's basically the filling in of everything. Its functions is to support, defend, transport, storage, repair. Characteristics. Its function, like, it varies in its constituents, but it has a larger amount of extracellular matrix. And we didn't discuss extracellular matrix at all when we were talking about epithelium. Extracellular matrix is made out of fiber and ground substance. And now, why did I not discuss that? Because epithelium cell doesn't have a lot of extracellular matrix. Meanwhile, connective tissue has an abundant amount. So again, tissues made out of, they are like the tissues, by definition, they're made out of cells, specialized cells, and extracellular matrix. It depends on the ratio, and it depends on the cells. Then we have different kinds of tissues. Epithelium doesn't have a lot of extracellular matrix, but connective tissue has a lot. So we're going to discuss fibers and ground substance. And extracellular matrix is a continuous dynamic state that is constantly being remodeled, constantly synthesized, degraded, and this is happening by metalloproteinase, right? Keep that. This is how we classify connective tissue. We talked about its function. Let's talk about classification. We're going to see its composition. We're going to see how it's organized. So is it embryonic, cartilaginous, adipose? And then how is it organized? Is it loose or dense? Is it regular or irregular? Now, we're not going to talk about all of these. We're only going to talk about embryonic. And we're going to talk maybe you're going to see, but we're going to talk about the organ. All right. All of these will be discussed later on in their respective blocks. For example, bone and cartilaginous will be discussed in MSK. When we're talking about tissue, we should know the cells, and let's talk about the fibers and extracellular matrix. These are the cells that make up connective tissue. We can classify them into two classes, or like two groups. Something called resident cells, something called wandering cells. From the name, resident, they don't move around. They stick to their place. Wandering cells, they move around, go up and down, go through the blood and so on. Hello. Resident cells, we have fibroblasts, adipocytes, mast cells, macrophages, adult stem cells. I'm going to discuss all of them except for adult stem cells. Wandering cells, we have plasma cells, lymphocytes, basically your leukocytes and monocytes. Tamam? Hello. It's important to note that a monocyte is the same as a macrophage, right? But monocyte is my wandering cell that then turns into a macrophage when I need it to. And the macrophage doesn't move around. And all of these cells are responsible for immune responses, right? These are very powerful cells. I don't want them everywhere. I don't want them to wreak havoc everywhere, especially the macrophage. The macrophage is one of the strongest cells ever. So it's, subhanAllah, it's a resident cell. My monocyte will go exactly where it needs to be, and then it's going to turn into a macrophage. Well, this is just extra information, by the way. I just like to talk. Now, when we talk about fibers and extracellular matrix, fibers components, like if the connective tissue, they're elongated structures. They basically proteins that are elongated and they fill in the matrix. We have three types: collagen fibers, reticular fibers, and elastic. I have some questions. What's your main thing after this? This is the last thing, all right? Let's talk about the cells first and then fibers, and then we're going to look at different types of connective tissue. So starting with fibroblast. Now, you guys should identify the cell by how it looks. Fibroblast, all right, it has a fusiform shape and it's elongated. So So it's flattened out, and around it you see a collagen. How does collagen look? It looks like this pink. We're gonna see it again later on. Remember all the times I told you guys about collagen type 4, type 3, type 1, type 7, 17. Okay. Now we're gonna discuss collagen, all right? هلا fibroblasts, their main function is to produce and maintain most of the tissue's extracellular components. They synthesize and secrete extracellular matrix. What is making up extracellular matrix? Fibers, ground substance. So my fibroblasts are making my collagen, my elastin, my fibrillin, and they're also making my ground substance, glycosaminoglycans, proteoglycans, and glycogen. I'm going to talk about ground substance in a moment. Let's finish this out first. Adipocytes are basically just cells filled with lipid inclusions, right? No one's talking. Is there any place where we can access the presentation? All right. If you go to your batch group chat, the 18th batch group chat, and if you open the description, you can find in the description a link called GND 2526 slide drive. Over there, I'll upload the presentation. You're going to find everything, right? And we're going to upload it later on with the YouTube videos. So, adipocytes, they are mainly one large droplet of neutral lipid. So this is entirely just one large cytoplasmic inclusion. And usually the nucleus is on the peripheries, all right? So this is still a live cell. Where do we find this? What's the functions of fat? Or like adipocyte, let's say. It's going to store my lipids for me. It's going to use my lipids for production of heat. It's going to cushion my tissue, and it's going to insulate my skin. Easy peasy. Cells. These are important, crazy cells. These are lunatic cells, all right? You don't want to activate it unless it's an emergency, unless we have an immune response. So they're oval, irregularly shaped. We have a lot of basophilic granules. Can you guys see this? And I believe if you guys took your hematopoiesis lecture, you guys understand now what basophilic granules are. In mast cells, you have so much basophilic granules that it's even including the nucleus. So it's a very easy cell to identify. Just a cell with a lot of purple dots. What are these basophilic granules? They are mediators of inflammation. Histamine, heparin, serine proteases, chemotactic factors. You don't need to know what they do. Just know what they are or where they're from. Histamine, heparin, serine proteases, chemotactic. Right? When you study immunity, you can understand it. Macrophages, I already talked about this a little bit. It's a differentiated monocyte, and sometimes we refer to as histiocyte. There are certain diseases, other things, all right? This is just a fun fact. And macrophages vary a lot in shape, all right? Like sometimes it's very easy to identify, sometimes it's not. Refer to your hematopoiesis lecture because you're going to have better pictures of macrophages there. But you should know their function. Macrophages phagocytose, debris and pathogens, and also they're responsible for tissue repair. So we're going to see them in areas abundant with inflammation. Plasma cell and leukocytes. These are your wandering cells, and these cells are your immune response cells. A plasma cell is the antibody production cell, all right? And they say clock face appearance, like I'm not going to lie to you and say I do see it. Personally, I don't see it, but I'm not telling you just information. It's a buzzword. Sometimes you see in a question, they say we took a histology slide, you can see clock face appearance. Plasma cell associated, all right? So if you do see it, happy for you. Antibody production, all right? And leukocytes, you guys have an entire lecture on that. Their main function is the immune response. They vary. You have many different kinds, eosinophils, neutrophils, so on, all right? Not focus on the fibroblasts is a form of fibroblasts. So we have the word fibroblast and myo before it. What does myo mean? Muscle. So it's a fibroblast. It's an activated form of fibroblast, and it has a lot of actin and myosin because it's similar. It has like similar behavior to a smooth muscle cell. And it takes part in repair and also just the entire wound healing process. So whenever I have an injury, I have a cut or anything, my body wants to repair itself. It's gonna call out fibroblasts, differentiate into myofibroblasts, and then I'm gonna carry out wound healing. Gonna be a big focus in second year. For now, just know basic idea. Collagen, everyone's favorite topic, I think, by now. Collagen fibers, here's some facts about collagen. So, the most abundant protein in the human body, and it's the most abundant structural component of connective tissue. We said earlier, it looks like pink and so on. So, over here, I can see fibroblasts, and all of this is collagen. تمام؟ هلا, collagen, it's a protein, and its main function is support. High tensile strength for support. The way it's made is important because in that process, there could be various variations that give us the different kinds of collagen. How is it synthesized? It's 3 alpha chains forming a triple helix. This helix, then, we call it procollagen, will turn into something called tropocollagen by an enzyme called procollagen peptidase. So, later, procollagen becomes tropocollagen, collagen fibrils, collagen fibers, like they're all going to bundle up and compile into, lastly, collagen bundles. We can see here our collagen bundles and fibers. هلا, this process gives us and allows us to open up so much possibility for collagen. We have passive types. Passive associated with the function, types associated with the structure. The classes and the structures and the types, do you need to memorize everything? Not really. But what you should know is the things we already talked about. Let's go through the classes real quick. We have fibril forming, we have network forming, fibril associated transmembrane collagens. Maybe these are just headers. You don't need to understand anything. But we took network forming. We took type 4 in the basement membrane. We know this. Fibril forming collagen type 1. We know that. Now you know. All right? Hala. When we talk about types, we have type 1, 2, 3, 4, 5, 6, 7. Like if there's 17, there's a lot. Do you need to know all of them? No. No, and focus on what we mentioned. So we mentioned type 3, we mentioned type 4, we mentioned type 7. Right? So type 3 is reticular, it's part of the reticular lamina. Type 4 is basilar, it's part of the basilar lamina. Type 7 is anchoring type 3 to type 4. Now I want to add on top of it type 1 and type 2. And hyaline collagen and elastic cartilages. And this form of collagen, you'll see it again and again in MSK and in the trachea and a lot of places. So type 2 is hyaline collagen. Keep that. Type 1 is like your fibril, cartilage, dentin, cement, and so on. Type 5, not really. We finished collagen. Easy peasy. Not that scary beast. تمام. هلا reticular fibers and then elastic fibers, and then we're going to take a look at connective tissue, and then we're done. Like ground substance as well, but regardless, reticular fibers. You guys remember how collagen looked like? It's very dense, it's very woven into each other, it's very parallel and everywhere. Reticular fibers, from the word reticular, they're mainly collagen type 3, and they're branching out everywhere. So in a form, this is my own naming, they form like our own batch naming, cherry blossom like branching. Come on. Stained black with silver sauces for your info. And before discussing the function, let's talk about where we find them. The location of reticular fibers, they are lymphatic and hematopoietic organs. Basically organs with a lot of ins and outs and communication. So it's in a sense exchange of content. What's this content? Cells. I'm exchanging red blood cells, mature blood cells, immature blood cells. I'm exchanging white blood cells and so on. So I'm doing this through this network, which is my reticulum. Elastic fibers, the third type of fibers, made out of elastin. And elastin has rubber-like properties. And now, this time around, I'm not going to go to the location. I'm going to talk about the function first. It's going to, through elastin, through its rubber-like properties, it's going to withstand stretching or bending. Where in the body am I going to have stretching and bending? Areas with a lot of pressure changes, such as in the lungs when I inhale and exhale. Air goes in, I need to expand my lungs. Air goes out, I need to retract my lungs. Regular stretching and coming, retraction, everything, right? And my blood vessels, my aorta, my blood pressure, my arteries, the standing and contracting and stretching, all right? All kinds of stuff. I need to be able to withstand this, and I do that through elastic fibers, through elastin. Now, this is my extracellular matrix. This is my connective tissue. My resident cells, you can see a macrophage, adipocytes, fibroblast. You can see my extracellular matrix, my elastic fiber, collagen fiber, reticular fiber, everything. Okay. Now let's briefly talk about ground substance. And then we're going to move to connective tissue. Sorry. Ground substance is my other component of extracellular matrix. So we said fibers and ground substance. Ground substance is made out of four things. Water, which we call also the interstitial fluid, glycosaminoglycans, proteoglycans, and glycoproteins. What are these three? These are the big bads right now. I think you guys are all scared by them. I believe you guys will also take a more lecture about them, so don't worry. We're gonna give you a good foundation on the function of ground substance. Again, it's filling out the extracellular matrix, and it allows diffusion of small molecules. These are for your info. Now let's talk about GAGs, proteoglycans, and glycoproteins. Glycosaminoglycans, shortly GAGs, they are basically, okay, firstly, all of these have the same function more or less, like diffusion of and communication of molecules, and they form extracellular matrix. The way they differ though is by their structure. Let's take GAGs first. They are repeating disaccharide units. So they are a carbohydrate. Now we have a lot of GAGs, like a lot of different kinds of GAGs. The most common and ubiquitous GAG found in almost all cells is hyaluronic acid, hyaluronan. And you guys should memorize this. You guys should know this. Like GAGs also make up proteoglycans. So we have these four GAGs right here: dermatan sulfate, chondroitin sulfate, keratin sulfate, heparin sulfate. These form proteoglycans. What is a proteoglycan? Proteoglycan. Now the structure is a protein polysaccharide complex. First part is the protein, it's the core structure, and then I'm gonna add to it the glycan, my GAG. If you remember, my GAG it's a disaccharide unit, it's a polysaccharide. So my core protein, perlecan, syndecan, lomecan, aggrecan, I'm gonna add to it these four GAGs, whether it's dermatan, and I'm gonna form my proteoglycan. This is a headache. Do I need to memorize all of this? Are you telling me I should memorize all of this? No. An easy way to memorize this. Proteoglycan. So these are the cans. If you see a can, it's part of the core protein. And if you see a GAG, it's going to be a sulfated GAG. So our GAG here is sulfated, and my protein core is canned. Lastly, glycoprotein. So it's a polysaccharide protein complex. It's the other way around. Does it matter? Absolutely. If I'm starting with a protein and I'm adding onto it a GAG, it's much different than if I'm starting with a carb, adding onto it a protein, all right? These are two different things. They interact differently with the environment and a lot of things, all right? So, glycoproteins, we have our polysaccharides and protein complexes, fibronectin, laminin, and they provide adhesion for epithelium cells. This should be fine. Going to memorize GAGs, you should know hyaluronic acid by heart. Any sulfate is a GAG. The way to memorize proteoglycans, any can is the core protein, and the sulfated GAGs are part of the proteoglycan. And glycoprotein, just memorize fibronectin. This is the full picture. Substance, collagen fibers, elastic fibers, blood capillaries throughout, fat cells, mast cells, macrophage, plasma, you call it. It's all everything. And then inside my ground substance, I'm going to have my water, I'm going to have my proteoglycan, GAGs, and glycoproteins. And on to our final three slides, I think. Now let's talk about connective tissue. We said we were going to classify whether it's dense / loose, whether it's regular / irregular. Like here, I have dense, irregular connective tissue. Like connective tissue, meaning it's collagen fibers. Maybe I have some elastin, maybe I have some reticular, whatever it is. Let's say all of this is collagen, all right? This collagen here is loose. It's dense, but it's irregular. It's dense because there's a lot of it, but it's not in one orientation. Some of it's going hick, all right? It's not in one orientation. No definite orientation. So we call it irregular, but it's dense. There's bundles and bundles of it. Main function, resistance to stress from all directions. For example, in my skin, I can go hick. I'm gonna have the ability to withstand this stress thanks to my dense irregular connective tissue. So it's located in my deep dermis layer of the skin and the capsule surrounding most organs. Dense regular connective tissue is the same story, but now we have an orientation. All of these fibroblasts are aligned in parallel and they're secreting this collagen in parallel. It's going to be the strong parallel connective tissue. And it's mainly collagen type 1, right? They're aligned in parallel and it's going to provide great resistance in the same singular direction. So, for example, tendons, neurosis, ligaments where I need that stress control and protection in a direction. Lastly, loose, we call it areolar connective tissue. And you can clearly see the difference in density. This is cells everywhere. Here we barely have any kind of cell, any kind of extracellular matrix, So it's mainly collagen fibers, and it's just like fibers, ground substance roughly, and it's just much less. Main function, similar to connective tissue, layer beneath the epithelial lining, filling the spaces. And you can find this again in the dermis, providing me this like flexibility rather than protection, and also in surrounding organs and GIT tracts. So these three different types, you should know how they look and how they function. Then I have my mesenchymal embryonic and my mucous embryonic. My mesenchymal embryonic connective tissue, these are both embryonic, so we only find them either in the embryo or the umbilical cord. My mesenchymal is found in both, and you don't really need to understand their function, just know how they look and where you find them. And spindle-shaped cells, collagen fibers are present, gap junctions are present, so these cells are communicating with each other. My mucous embryonic is only found in my umbilical cord, and it's less dense than my mesenchymal. Like the cells are widely separated, they appear like fibroblasts, and also we call it Wharton's gel. It's the full picture. The more we go down, the more dense it becomes. This is loose tissue. This is somewhere in between. So this is loose areolar tissue. This is loose connective tissue. This is dense irregular. This is dense regular, and this is very dense, very, very regular. And with this, we conclude. We finish everything. Thank you all. And if you guys have any questions, feel free to ask. We just finished 6 lectures. We finished cell 1 and 2, epithelium 1 and 2, and connective tissue 1 and 2. And if you guys want to scan the QR code, feel free to give me feedback. Tell me what you liked about the session, what you would like to improve, anything. And yeah, thank you. Best of luck on your exams. Inshallah, inshallah, everyone does the best, gets the grade they want. And if you guys have any questions, feel free. Anything you want. Thank you, guys.